Good afternoon, and thank you for joining. I'm Matt Roth with Baylor College of Medicine, and I welcome you to today's presentation. The Extracellular RNA Communication Consortium is an NIH Common Fund program which works to advance the science and research of extracellular RNA. The consortium hosts monthly presentations on a variety of research topics, and I'm pleased to introduce today's speaker, George Calin from MD Anderson. His talk today is titled, Loading Cancer Drones, the Regulation of Exosomal Non-Coding RNA Content by TP53. George?
So thank you, Matt, and thank you, Tanya. It's very nice to be here with all of you, and thank you, everybody who is attending. So basically, what I will do here today, I will tell you about our work that started about 6-7 years ago on the concept that the most important tumor suppressor gene in the human genome regulates microRNAs, and how we move this one from inside the cell to outside the cell, and how we move from short 21 nucleotides non-coding RNAs to longer human-specific transcripts. So I will put together a story in which I will show you how size doesn't matter. This is something strange for Texas, to say that size doesn't matter, but I will tell you that short or long non-coding RNA doesn't matter, they have a big impact. And I will link this concept to the one that cell-to-cell communication involve a new type of hormones. And these new type of hormones are short or long non-coding RNAs. Now, this is a concept that it's very much told here and there these days, but think in a very simple way. Tom Czech got a Nobel Prize years ago, which was in absolute one of the quickest Nobel Prize after the initial discovery, on the concept that the RNA world was before any DNA or protein world. Now, the ribozymes identification was basically one of the most unexpected, and what taught us is not only that the RNA world was initial, but genome-to-genome communication in a world full of RNAs probably involved small RNAs, because there are no DNAs and there are no proteins surrounding. So conceptually, we can say that short RNAs like microRNAs should be one of the oldest hormones who communicate between different cells. And let's tell you how the story starts. The story starts by focusing on this very little genes named microRNAs, and now is no need to tell you what microRNAs are, because there are over 50,000 papers in about 10 years published on microRNAs. But in 1993, when they were discovered, or in early 2000, when the boom of microRNAs started, it was really a big deal. So microRNAs are these very short 21 nucleotides transcripts. Here I show you the pathognomonic point of identification of a microRNA, which is transcribed as a hairpin structure. And what was supposed to do these microRNAs? They were supposed to target 3' untranslated region of protein coding genes and knock down the expression of important proteins for any type of mechanism in quite any type of cells from vertebrates. Now, where starts the story that I'm working? Start on chromosome 13. Now, I'm Romanian. I play a lot of soccer. We never play on 13 because our team is losing forever. So I got coming in a 40+ people laboratory this project to identify what are the genes important in a specific deletion found on chromosome 13 in the most frequent type of Western leukemia, the chronic lymphocytic leukemia, but also in multiple myeloma, prostate cancer, and pituitary adenoma. As you can see, here are 3 different regions of deletion that the people were identifying, but no gene was found in that area. Not only, but when we start cloning day and night these genes, we cover ourselves of shame, because in early 2000, to tell that you clone these red genes, which means transcripts who have no open reading frame and no putative protein was the meaning that you are a poor cloner. You induce mutation, insertion of deletions, because the dogma was each transcript should codify for a protein coding gene. But luckily, we identify that two very small RNAs, named microRNA 15 and 16, are located in this region of deletion, and from here start the story of microRNA involvement in cancer, and I can say in any type of disease, and the involvement of any type of non-coding RNA in any type of disease, by the simple identification of two small RNAs in a region of the genome where everybody was looking for decades for important genes. Now, I don't know how many people are now attending from the trainee, but if it is a student or a postdoc in the people who attend, pay attention. Never think that where the papers are telling you should be an important gene, it's nothing because the literature or the main concept, scientific concept in that moment did not fit. In early 2000, nobody believes that microRNAs are really important. 1993, they were discovered in worms. In 2000, first, let's say, one family of conserved microRNA was identified. So they were considered these short genes like something, an anomaly of the genome. Look, they are present where you are expecting to be present important genes, and from here starts the story. And what I show you here is something that was very surprising for us. The fact that in this most frequent type of leukemia in the Western world, if you analyze all microadenoma by a genome-wide technique, in that moment, 2005 was array, profiling by array. You identified about 13 genes, again 13, my lucky number. Out of this, MIR15 and MIR16, which were located in the most important deleted region, which these 13 genes perfectly were differentiating patient with good prognosis versus patient with bad prognosis. Why CLL is a very interesting disease to investigate? It's because the patient either survive 5–10 years, which is a very good overall survival for a type of cancer, either about 10 to 15% die in less than 1 year with a very aggressive type of CLL, named Richter transformation, that do not respond to any type of treatment, including the newly gold standard ibrutinib, BTK inhibitor. And these patients in less than 1 year are dead. Now, this is an overall survival much poorer as the one for pancreatic cancer, for ovary cancer, or for glioblastoma, which are the pathognomonic cancers, so-called deadly cancer with a poor survival. What we have done, it took us about 6 years to identify why 4 of these microRNAs are important in survival of this patient. And what we identified a quite complex network of short RNAs, microRNAs, and protein coding genes, which are located in the 3 most important deleted region in CLL genome: 13q deletion, microRNA 15/16, 17p deletion, p53, and 11q deletion, microRNA 34b and c. I don't want to tell too much about these things because they are published data, but what I want to point out is the fact that when the patients had CLL with lost 17p, they were basically the people who were dying in less than 1–2 years, because p53 knockdown was basically activating MIR34, was basically reducing the expression of MIR34B and C, which was activating a very bad kinase, ZAP70, which is doing a lot of harmful in the CLL cells. Second, if the patient were losing chromosome 13, these patients were surviving very long. Why? Because MIR15/16 have a targets BCL2 and MCL1. Low MIR15/16 means high BCL2, high MCL1, which are basically anti-apoptotic protein coding genes. So these malignant cells were surviving very nicely, very long time. Now, going forward here, in the same time, in parallel with this identification of several microRNAs, who are regulated by p53. We make in collaboration with a very good and very bright scientist and young scientist, Muller-Fabry, in University of Southern California, unexpected findings. It is a microRNA, the first ever microRNA linked to be oncogenic in vivo, microRNA 155. Carlo Croce's group published a PNAS in 2006 showing that overexpression of miR-155 induced in mice, transgenic mice, induced acute lymphocytic leukemia. What the people were publishing, and you can check the literature of over 2,000 papers on miR-155, miR-155 is very poorly expressed in epithelial malignant cells. Also, a lot of publication shows that this microRNA is overexpressed in tumors when you are doing the profiling. So either these profiling data are all wrong, either this microRNA is not expressed in the epithelial cells, but it's expressed in tumor microenvironment cells. So around 2010-11, we started this collaborative project in which what we have done, we profiled exosomes from malignant cells from children with a very bad type of cancer, neuroblastoma. And what we found, it's simply shown in this cartoon. So pay attention. MicroRNA 21, which is in absolute the microRNA which is overexpressed in any type of solid and malignant cancers. If you do profiling of any cancer, one of the top highly expressed microRNAs is miR-21. miR-21 is produced in the malignant cells. It's released in exosomes. These exosomes go according to the cell-to-cell communication hormone theory in tumor-associated macrophages. miR-21 is binding to the toll-like receptor as an agonist, which is a very interesting finding done by Muller-Fabry in parallel with a German group, the fact that microRNAs are not only targeting messenger RNAs directly by sequence complementarity, but they are also agonists of toll-like receptor, a completely independent and a new function of microRNA. MicroRNA 21 targeting toll-like receptor 8 induce NF-kappa beta, and NF-kappa beta is known to induce what microRNA? MicroRNA 155. But this didn't happen in the malignant cell, happened in the tumor microenvironment cell. miR-155 is getting inside the exosomes, but not the exosomes delivered by malignant cells, the exosomes delivered by tumor-associated macrophages. And go where? Go back inside the malignant cell. And this is why you identify some level of miR-155 in the malignant cell, because this is produced not by the genome of the malignant cell too much. It's produced by the genome of tumor microenvironment cells, which release miR-155 inside the exosomes. Going back to the malignant cell, where what is doing? It's targeting a specific protein coding genes named TERF1, reducing the expression of TERF1, increasing the expression of telomerase, because TERF1 it's an inhibitor of telomerase activity, and this is linked to drug resistance. So why I was spending 2-3 minutes to explain you this step-by-step mechanism, because this paper and this experiment, this long time of work on this manuscript told us something. The fact that exosomes circulate very important microRNAs for resistance to therapy in cancers. Now, what is killing the patients? The patients are killed by the widespread dissemination of malignant cells who do not respond to chemotherapy. So from here start the project that basically we had we were very lucky now to get these funds from NIH and to push forward on this project, in which what was our goal? Our goal was basically to link the identification of a p53 network of microRNAs that I told you initially was published in the JAMA paper, with this GNCI identification of the same microRNAs from this signature being present in the exosomes. What was very important was that we identify this one in 2 different types of cancers, meaning that the mechanism can be a mechanism which is general in multiple types of cancers. And look what surprisingly we identified. So we start with 3 different models of p53 deregulation. First, it's the REH, which is an acute lymphocytic leukemia cell line, because high 155 mice develop ALL, so we move on this type of cancer, and we knock down p53 in these cells. Second was a SCOV, which is a ovarian cancer cell line, and the normal HEO108, which are the cells, the normal epithelial ovarian cells. And the third is the classical, I can say already, HCT116 colon cancer cells with the shRNA for p53, which knock down p53 as in REH, and we use also the now famous Vogelstein model of HCT116 cells with deletion of the first exon start codon, so the p53 protein start codon is not present. Now, we purified the exosomes with the ultracentrifugation method. We check if the exosomes are present, and in fact, in the image in the right side, you can see here, this is a bar of 100 nanometers. You can see these vesicles who are around 100 nanometers, so they are of the exosome size, and also markers for exosomes present when we have done the westerns. Now, what was immediately surprising for us and what we have done, we test miR-155 in these exosomes who come out from p53 knockdown cells, and we identified that both 5' and 3', 5p and 3p 155 are overexpressed in the exosome from the cells where p53 expression is knocked down. And from here we start a full story that is now in press in clinical cancer research, so I will, it has to come out shortly, so I can tell you which is a story, a therapeutic story. I didn't tell you before, but my job was as gastroenterologist and intensive care unit physician, so I still believe that what we discover as a mechanism, we have to move as soon as possible to the clinic, because we like or we don't like, after probably hundreds of billions of dollars spent in research in the last 20, 30 years, the mortality by cancer is slightly decreased in the last few years by the simple fact that the people smoke less. Lung cancer mortality is decreased in a decent way in the last 3-4 years by American Cancer Society reports due to a very simple habit: do not smoke. So a lot of research still did not produce the breakthrough in order to induce a significant reduction in the mortality of cancer patients. So look what we found. We found a pathogenetic model in which miR-155 knocked down p53, and here we identify by using different targeting softwares in 3' UTR of p53, different binding sites for miR-155. We have done what everybody is doing, luciferase assays. We mutate this site, and we identified, in fact, that miR-155 has a binding site in 3' UTR of p53, and when we use lung cancer cells and inhibit the miR-155 level, p53 levels are higher. And here you can see better in two different cell lines where we use the anti-miR-155, not anti-miR, we use miR-155 mimics, the level of p53, p53 inhibitor protein 1, and p21 are going down, meaning that miR-155 regulates p53 by direct interaction, and very important, the downstream network of p53. Now, not to make the story very complicated, we identify that p53, not only p53, but also p53 inhibitor protein 1, it's a direct target of miR-155, something that was known before. So the meaning is 155, it's targeting p53 and other important protein coding genes from p53 pathway. The second part of the negative feedback is that p53 is a negative regulator of miR-155, and we identify again by bioinformatics in the 5' region of the the pre-miR155 in the genome binding site for p53, we have done the promoter experiments, and in fact, what is, making a long story short, is that p53 negatively regulates 155, 155 negatively regulates p53, and again, keep in mind, this 155 is present in most of the exosome in a high level exosomes that circulate between cancer cells and tumor microenvironment cells. Now, here starts really the interesting point. What we identify by doing experiments in multiple type of cells, including ovarian cells, here I show you lung cells and ALL cells because the paper is focusing in this moment on this type of cancers. We identify that the cancers with high level of 155 in the cells are more resistant to chemotherapy, but more resistant to multiple types of chemotherapy. So doesn't matter you have cisplatin as treatment, doxorubicin as treatment, ovarian cancer, lung cancer, triple negative breast cancer, chronic lymphocytic leukemia, acute lymphocytic leukemia, the cells with high miR155 are poor responders to chemotherapy, multiple type of chemotherapy. So what we identified, by good luck, of course, we identified the fact that miR155 and p53 represent a central network for resistance to different type of chemotherapeutics. What we generate then we generate in collaboration with Bulent Osbolat and Gabe Lopez-Berenstein, who are Gabe is the PI of this UH2, UH3 with Anil Sood and me. So we create some delivery vectors who have on the surface some changes which make them to go specific to the tumor cells and to the tumor microenvironment cells. So thinking to the anti-miR155 therapy, which you remember, miR155 is generated in the tumor microenvironment but act on chemoresistance on the malignant cells, we generate this double sword type of delivery systems which are going to epithelial cells and tumor microenvironment cells. And we took, of course, multiple models. One, what that I show you here, it's a model of cisplatin treatment of A549 cells with high miR155. A549 are lung cancer cells. And to our surprise, when you combine cisplatin with anti-microRNAs in this specific type of delivery systems, here at the right column, basically the primary tumor size is quite zero, while anti-miR155 treatment alone or cisplatin treatment alone did not reduce so much the tumor size. So this is very important. What is the meaning? The meaning is that the clinician can continue to use chemotherapy as they are used for the last 40 years now. Frey and Freyreich at the end of 60s discovered, when they were working at NIH, discovered the combined chemotherapy for children with acute lymphocytic leukemia. They were very much criticized that they will kill the kids because they put together 2 or 3 chemotherapy drugs already toxic, and they didn't kill the kids. They transform a deadly type of cancer in a cancer, very curable type of cancer. Children with ALL are surviving now very well, and they are cured simply by combining chemotherapy. So the clinician will continue to use this type of chemotherapeutic combination, but what we propose that they will add minimal doses of anti-miR155, which are not so harmful like single anti-miR therapeutics, because you give to the patient lower doses because this is an adjuvant therapy, and at least in mice we were able to wipe out the malignant cells. Here I show you that this is linked to p53 levels. We have done the in situ hybridization for p53, and in fact, when you put anti-155, the level of p53 are very much down. So meaning this effect, it's in part related at least to p53 network. And then we move forward to triple negative breast cancers, where we have done a treatment with doxorubicin using MDA-MB231 cells, which have high level, endogenously high level of miR155. And again, when you make the combination doxorubicin and anti-155, the tumors are quite disappearing. So basically the tumor weight, it's either zero, either very, very low. And here we identified in the histological stains of the tumors that we collect from mice the fact that the combination anti-155 and the chemotherapy, you see this column, has the lowest number of Ki67 positive cells, so the lower number of proliferative cells, has the lower number of CD31 positive cells, meaning the lower number of cells who produce blood vessels, so the lowest neogenesis vasculature, and has the highest level of tunnel positive cells, highest level of apoptosis. This makes sense. So if you add anti-155 by targeting p53 pathway and other pathways that we present in the paper, but I don't tell now because it's not too much time, to the chemotherapy, the cells are dying the most, the cells proliferate the less, and the tumor, because miR-155, it's knocked down not only in the malignant cells, but also in the tumor microenvironment cells, the tumor is not producing enough blood vessels in order to keep the cells alive. Now, very important on the light of the failure of miR-34 substitution in individuals with advanced type of cancer. So miR-34 by miRNA Therapeutics trial was stopped because of severe immune reactions. Now, very important for this type of therapeutics, we have done a very, very deep and repeated a couple of times dose toxicology effects. So as you can see here, the most enzymes which show the liver function, the kidney function, did not change significantly. Not only, but the white blood cells, red blood cells, and different type of blood cells, they did not change significantly in the mice with scramble treatment versus the mice with 155 treatment. I agree with you. This is in mice. Now, of course, we have to move forward with primate animals experiments, but still, this is a necessary step for all IND requests. Furthermore, a large panel of 23 cytokines shows that except few of them, MIP1B, 1 alpha, and interleukin 17 and 12, all other have different changes but not significant. And furthermore, staining of multiple tissue show no macroscopic differences between the scrambled and anti-miR-155. We took brain, heart, kidney, and so on, multiple other tissues. So making a long story short, what we propose here? We propose here that by simply analyzing the extracellular RNAs, we identified miR-155 as an abundant microRNA in exosomes, and also, now I can tell you, in plasma, independent of exosomes. Probably miR-155 circulates also in lipid rafts, is bound to proteins, or circulates in other type of vesicles. But it's an ubiquitous exogenous RNA that is produced by tumor microenvironment cells mostly, much more as by the genome of the malignant cell. And if you knock down the production in the tumor microenvironment and you knock down the amount of miR-155 who go in the malignant cell, you can basically reduce significantly the chemoresistance to multiple type of chemotherapeutics in multiple type of solid and hematological cancers. I don't want to say this is a universal panacea, but I want to say that in respect with other type of microRNAs or drugs which are working in one type of cancers, we identified one type of microRNA which, when it's knocked down, reduce resistance without significant harm for the animals in multiple type of treatments. Now, this is one part of the story which everybody loves, everybody looks microRNAs, 50,000+ publication. But in early 2000, John Matic was writing very interesting reviews, and you can go in PubMed and check if I'm right or not, telling a big truth. But it took us about 10 years to realize that he was right. We were all looking to microRNAs. They were just identified, in 1993, there was a string of 3 papers in Science 2001. Everybody was realizing that microRNAs are not broken transcripts without any effect. They are very important transcripts which are produced independently of RNA degradation. But Mati was saying the genome is full of something that nobody was looking too much. Also, the people knew about this type of long non-coding RNAs from bacteria from early 80s. If you check on PubMed, if you compare the publication using the word long non-coding RNA versus the word microRNA, you will be surprised. Long non-coding RNA publications were at a rate of tens, if not hundreds, in 80s and 90s, but nobody was looking too careful to these genes. Why? Because again, the concept was one transcript, one protein. These are stuff from bacteria. Who cares about bacteria? Only the poor people with septic shock, which got infected, but otherwise, bacteria are something that is not very important for us, which is a very, very wrong concept. Look now how nice the field of microbiota is moving forward. So coming back to this story of long non-coding RNAs, I will tell you another point. The genomic dark matter that matter, but matter also in the exosomes. So what we have done, we move back to a very old concept that when you don't understand something, you have to read from the field that is outside your interest. So Noam Chomsky, as you know very well, and I wrote here, is the father of modern linguistics. He is basically the person who put down the way in which we think the languages are grammatically structured, and not only, but as you know very well, the C programming of computers and so on are based on what Chomsky was built years ago, and this is basically the way in which the computers are working. And he's also a person with very strong opinions. I don't want to talk about Chomsky, the politician or the philosopher, but I want to talk about the fact that his context-free grammars, telling that everything that we are talking has a specific structure, are basically important for putting together the sentences in which non-coding RNAs and protein-coding genes are together. I show you now a sentence in which miRNA 55 is central with another gene, which is a coding gene, p53, for treating patients with cancer. I will tell you here something which is, in my opinion, more exciting at this, because it hit something that is very important. What is the fundamental difference between cancers in humans and cancers in non-humans? And what we have done for here, for getting this, we took this deep sequencing data that everybody sequenced from RNA, and we compare with DNA motifs. So what we learn from Chomsky, the concept that patterns are very important. Patterns in how we are talking, patterns in how we are doing any activity on our life, and patterns in DNA. So on this cartoon, what I show you by different colors, different patterns in the DNA. What we were thinking, starting again from microRNAs, is the fact that where these patterns are transcribed like hairpins, this is a flag for a DNA location of a microRNA, unknown microRNA. And you can see here now the PNAS paper in 2015 by Isidor Rigoutsos, a bioinformatician now from Thomas Jefferson, who identified thousands of new microRNAs by doing this pattern discovery, and most of these microRNAs were primate specific. So what we have done, in 2005, 2006, I was much younger. I was still reading papers, you know, and I was reading a paper about published by this very interesting and very clever bioinformatician Isidor Rigoutsos, in that moment at IBM Research Center in upstate New York. And what he identified so-called pygnons. Now, pygnon in Greek means very many, very dense, which are DNA sequences of 16 nucleotides, which are repeated at least 40 times in the human genome, in the intergenic and intronic regions, and one time inside coding genes. So my understanding was that the most instances of the pygnon in the human genome are intergenic. What I have done, I checked these motifs. These are the motifs that Chomsky was talking. You see, 16 to 20 nucleotides repeated multiple times in the human genome, but never present in mouse and rat genomes. So these are human/primate specific motifs. Very interesting. There are about 225,000 pignons in the human genome, repeated tens of times. So the genome has millions of places which can flag, what do you think I was believing, flag human/primate specific non-coding RNAs. Why? Because they are present mostly in the intergenic and intronic regions. What we have done, 2008-2009, exactly because deep sequencing is very difficult to identify so many instances together, we prepare an array. A simple array done with, in that moment, NimbleGen, now done with adjuvant, an array in which we design probes, sense and anti-sense to the DNA pignon region, and also we put microRNA probes. And what we found was super surprising. We found that this region of the genome, again, hundreds of thousands of regions, are not silent regions, are transcribed as non-coding RNAs. And these non-coding RNAs differentiate normal tissues, but very important, differentiate perfectly CLL or colon cancer cells from normal colon and normal lymphocytes. So this is extremely important because what we identified that human-specific transcripts can profile perfectly human cancers. Furthermore, we pick one of the pignons that we name it enabler, and I will tell you shortly why. And what we have done in situ hybridization because we want to use a second technique that at least I, as a former clinician, I'm much more used to analyze the pathology and the radiography data as the arrays. And what we found that this pignon, it's much expressed in adenocarcinoma versus normal tissues, but please be very careful here. This is a hematoxylin eosin staining. This is a tumor site. This is a tumor microenvironment. This pignon, it's expressed specifically in the epithelial component of the cancer and not in the mass. This is the muscle cells from the tumor, or here in this place here down that we cannot see, there are tumor microenvironment cells. Furthermore, we analyze tens of such samples from a tissue microarray, and it's clear that this pignon, which is again a human or primate specific, in this case it's a primate specific long non-coding RNA of about 850 nucleotides, it's specifically overexpressed in adenocarcinoma, in the metastases from adenocarcinoma which are located in liver or in other tissues, but it's not highly expressed in benign or in inflammatory tissues. So this is a epithelial cancer specific expression. Then we have done what everybody in MD Anderson like to do. We make multivariate analysis with hundreds of colorectal cancer cases, and we found that multiple pignons, including others, are independently associated with the metastatic cases and with the metastasis, which makes sense. This is a long non-coding RNA overexpressed in metastatic cancers and in advanced cancers, so it has, if it is pathogenetically important, it has to correlate with a prognosis. Now, at this point, we took a step back and we look at what we know about non-coding RNAs and metastasis. And what we know, and it's very important, we know that in the metastatic process, many papers, also now this claim it's challenged by new papers, including from Raghu Kaluri's lab, that epithelial mesenchymal transition, it's important for metastasis. And not only in epithelial mesenchymal transition, everybody looks for microRNA 200 family. So what we have done, working in collaboration with Isidore Igoutsou now, very talented bioinformatician who produced RNA-22 microRNA interaction sites, we use this one, and we found surprisingly that this pignon 90 that we name enabler, it's targeting miR-141 and miR-200 from the miR-200 family. So what we identified, and this is a paper that it's in revision, so I will present you only some hints about the mechanism. We identify that epithelial mesenchymal transition and consequently metastasis are not controlled only by protein-coding genes like E-cadherin and ZEB1, not only by highly conserved microRNAs, but also by human-specific transcripts like enabler. This is very important because if you think when you check the literature, the most important mouse model of human cancer, the APC mouse model, which tried to reproduce the adenomatous polyposis colon mutation, a gene identified 30 years ago, which is one of the most important genes for colon cancer. These mice do not develop colon cancer. They develop small bowel cancer, which is a completely different disease in humans as large bowel cancer. So the meaning is, in the human genome should be something which creates the humanity fingerprinting for cancers. And we found this type of long non-coding RNAs. We have done correlations and we identify that in fact this enabler expression, which is not very high, it's a moderate expression, correlate with the E-cadherin levels in normal and tumorals. And we identified, as I told you before, that by in situ hybridization, as you can see here, these are the U6 expression. This long non-coding RNA, it's expressed at levels that are lower but comparative with the levels of housekeeping genes. So we were able to identify this one by doing quantitative PCR, by doing in situ hybridization at levels that are comparable with other coding genes. And very important, here it's only a snapshot, we deleted the 16 nucleotide pignon inside the 850 nucleotide transcript. In this region of the pignon, it's the region which is supposed to interact with, is predicted to interact with miR200 family. And in fact, we identify that you knock down this one, the miR200 levels are restored. And not only, but the relative matrix invasion, so the invasion and migration, can be basically modified if you knock down this region or you keep this region inside the transcript. So otherwise, the most important part for the interaction with miR200 from this super long transcript are simply 16 nucleotides motifs who are present in the genome, are transcribed in these transcripts, are named pignons. From here the name transcript pignons, and they are what are they are doing? They are hubs of interaction with microRNAs. These are data done in my laboratory, of course. I ask also Rigouso's laboratory to reduce this data in an independent way, and you can see here the deletion of the pignon is basically rescuing the phenotype. Here it's a colony formation assay that it's induced, it's increased with the wild type enabler, come back to the levels of the control vector when you delete the 16 MERS. But not this is the main point why I told you the story. I told you the story because here are a set of over 160 colon cancers in which we simply have done a lot of other things, but we simply check the question, how much are expressed pignons? Because in order to be buffers for microRNAs should be expressed at comparative level with microRNAs. Surprisingly, on the array which contain most of the known microRNAs, but only a part of the pignons, because we cannot put on array 250,000 pignons in the human genome. You can see here the pignon expression. It's several order of magnitude on a logarithm 2 more expressed as microRNAs. MicroRNAs are expressed at an order of 10, pignons at order of 16, so these are about 50, 60, 50 to 100 times more, meaning that in fact the human genome has a pervasive human-specific transcription, which is basically buffering non-coding RNA, which are more studied, shorter, and more conserved as microRNAs, very probably circular microRNAs, very probably other type of transcripts. What has this to do with Chomsky? It has to do because this discovery can be bound in a sentence. The sentence is enabler, a human-specific non-coding transcript, buffer miR200 family, ZEB transcription factor and E-cadherin, and modulate EMT. Has this something to do with the exosome and cell-to-cell communication? Yes, and here I will tell you a lot of unpublished data, which now we try to bounce them in a publication, and we come back to the story of p53. That was a story that we used therapeutically Basically, here are the models that you use only to remember you again. A leukemia model with shRNA against p53, ovarian cancer model where the HEO180 cells are the normal ovarians, they have normal p53, SCOP cells have mutation and deletion, a frameshift deletion with stop codon, so p53 genes, it's stopped at the 4th exon, and a colon cancer model, this is a wild type and p53 minus with the ATG codon 1 replaced by Fogelstein, and also we use another model of ACT where the same shRNA used for REH, basically it's used for p53 to have knockdown of the p53 transcript up to 10 to 20% of the expression. And here is the surprise. What we identified, I think it's really interesting and show how clever the malignant cells can be. So I will show you the data in two models, the leukemia model and the ovarian model. So what happened here? When you compare the level of different transcripts profiled by our array with the level from the cell of origin, you identified that when p53 is knocked down, the number of pignons, human-specific non-coding RNAs, it's higher when the p53 as the number of highly expressed pignons. So when p53 is knocked down, the amount of pignon from exosome is reduced significantly. Conversely,
George?
Yes?
Just heads up, we have about 12 minutes left.
Okay, I will finish. I have 5-6 more minutes. Then
okay.
The level of microRNAs are significantly upregulated. This is found also in the ovarian cancer model, cancer versus normal, in which not only p53 is abnormal, but the meaning of this very similar data is that p53 is a main regulator of this mechanism, in which when p53 is down, the amount of microRNAs, short RNAs very conserved in exosome, it's increasing, while the amount of long non-coding RNAs poorly conserved, human-specific, it's decreased. And not only this one, multiple other type of non-coding RNAs have different expression, like the ultra-conserved transcripts that I didn't talk because there's no time, or other long non-coding RNAs. I give you only an example like cyst or hot tip. What are the potential mechanisms? And here it's where we move forward. Years ago with Danilo Perotti, we identified a very interesting mechanism. Before Pierpaolo Pandolfi identified his decoy sirna mechanism, we identified that RNA binding proteins, a specific one, HRMP-E2, has a specific interaction with microRNA-328. So what we have done further, and here is the mechanism that we proposed in that Cell paper in 2010. What we have done, we analyzed the expression at the messenger level of the RNA binding proteins in our models. And not surprisingly, we identified that a lot of RNA binding proteins have differential expression according to p53 pathway. So what we are doing now, we are doing a lot of shift assays and interactor assays, thinking that what happened, it happens that the RNA binding proteins level are different when p53 is different, so they are shuttling different amount of small RNAs and long non-coding RNAs inside the exosome, because the level of these proteic shuttles are different. And we identify that the shuttles for microRNAs and the shuttles for specific human pignons are different. Furthermore, is not enough to show that messenger RNA levels are different. What we show that the protein levels are different by using proteomics with Sam Hanash and Ayumu Taguchi here at MD Anderson, and about 80 cell lines, we identify that specific RNA binding proteins have different protein levels according to the p53 levels. A second potential explanation is much simpler. It is not a problem of RNA binding proteins. It's a problem of p53 direct regulation of the short and long non-coding RNAs. So they are produced at different rates in the cells, so they are differentially translated in the exosomes. I put this one as a second potential mechanism because still, when you remember, our data were normalized with a cellular expression, and still pignons and microRNAs have a negatively correlated expression. But what I want to tell you here, I want to tell you here that against the concept that pigment transcription it's a random one, because it happened in hundreds of thousands of places of the genome, we have data showing that p53 regulates the expression of specific pigments, and this one, pigment 90, is the enabler. And we have done not only Natlin and expression experiments, but we have done also chromatin immunoprecipitation, and we identify the specific places where p53 is binding in order to regulate the expression, intracellular expression of these long non-coding RNAs. And finally, to finish, what has this to do with the phenotype? It has to do because these long non-coding RNAs are getting and short non-coding RNAs are getting into exosomes. And here I show you the ovarian cancer model in which the cell migration is significantly different when you treat you make the co-culture between the SCOF cells and the exosomes from the same SCOF cells versus no exosomal or versus the supernatant treatment. And this, it's normalized with the cell proliferation. So basically, it's not an issue of proliferation, it's an issue of migration. So this is an autocrine effect of p53 exosomes on the same type of cell. So making this second story short, we identify that p53 regulates differently the loading of short RNAs and long non-coding RNAs. And this loading of short and non-coding RNAs is related, at least in part, to the RNA binding protein mechanism. And finishing the story 10 minutes in advance, it's this one. What I want to tell you is not that the exosomes are important, not that microRNAs are important, not that long non-coding RNAs are important, but something more important, that the new El Dorado field of science, it stay in the non-coding RNA part of the genome. Non-coding RNAs, non-coding DNAs as super enhancers, insulator, modulators are extremely important. There are non-coding RNAs who regulate PD1, PD-L1, CTLA4. So immunotherapy is very much related also to the way in which non-coding RNAs regulates these checkpoint inhibitors. And finishing, here it's my lab. My lab last year, people coming from all over the world, and of course I have to thank them for all this work they have done, because without them we cannot give the talks. I have also to thank certainly to NIH and for these funds, and also to Anil Sud and Gabe Lopez, because we are 3 MPIs on this grant. And I have to tell you that in life you have to be very lucky for good mentors. I was lucky to have 3 mentors. Dragoș Ștefănescu is a mentor in Romania. I was born in a communist country, where genetics was forbidden because it was done by reactionary Americans. So in late 80s, early 90s, I learned about genetics from this professor, Dragoș Ștefănescu. Then I went in Italy in 97, 98, where Massimo Negrini teach me what is a pipette, because I didn't know. And of course, I have to tell you that among all the geneticists in this world, the one who believed from the beginning that microRNAs are important in cancer is Carlo Croce. And Carlo Croce spent a lot of money in 2002, 3, 4, 5, in order to push forward the microRNA work, and the non-coding RNA work, with no funding agency was paying neither 1 cent for microRNA work. You know why? Because microRNA were degraded RNAs without any importance. Of course, a lot of collaborators, and again, very important funding sources, because science, ideas without money are very cheap ideas, and you cannot put them into practice. So again, thank you very much for funding. Thank you very much for this time. And any question, if is no time for questions, then you can send me the emails at my MD Anderson email, gcalin@mdanderson.org. So thank you again to everybody for your time and for attending.
George, thank you very much. Very nice presentation. We do have a few minutes for questions, and people certainly can also email you. Any questions for George?
I did miss the first part of the presentation, so I'm not sure if what I missed. But I was just wondering if they have a sense of the difference between the different transcription rate and the degradation rates, because that can influence potentially how the RNA, especially the non-coding RNAs, how they're loaded into vesicles.
Yeah, you are right. So this is an important aspect. What I can tell you is that we didn't do this type of experiments yet. But what we have done, we designed the array in a way in which the long non-coding RNA have multiple probes located at a certain amount of nucleotides in between. So we are able to identify multiple multiple places of the same long non-coding RNA in order to claim if it is broken or if it is complete. And for several of them, we identify that multiple probes from the same long non-coding RNA light in the same way on the array. So the long non-coding RNAs load in the exosomes are not all broken pieces, are at least some of them full size. Your point is very good. In fact, we are doing experiments in order to see if it is a degradation or not, and if these rates of degradations are important.
George, on the feedback loop you showed with p53 and 155, are there any others involved in that loop?
Yeah, basically we found MIR15, MIR16, MIR34, and we found this new type of long non-coding RNAs, transcribe pigments, including enabler, to be regulated by p53. Certainly is not the only regulation. For example, we know now that also methylation regulates this new type of non-coding RNAs. So basically, there are multiple input mechanisms who induce a specific level of these non-coding RNAs, long or short. We focus on p53 because it's the most frequently deleted and inactivated tumor suppressor in the human genome. Also now, a lot of papers are talking about activating mutation in p53, which makes sense because p53 was identified initially as a non-cogene. It was not a tumor suppressor because the people cloned a mutated version which had an activating mutation. So the first clone of p53 was not the wild-type p53, was the oncogenic p53. But still, I believe p53 is a tumor suppressor, so it's knocked down and deleted in the most type of cancers. So this is why we were going to p53.
Mm-hmm. Thank you. We have time for one more question. Okay, well, if no other questions, again, feel free to email George. And again, this will be, it's recorded, it'll be posted on the portal at some point in the future after the publication comes out. And then we'll probably also have a blog on this. But thank you very much, George. And
thank you everybody, and thank you, Matt, and thank you, Tanya, and thank you to everybody who attended.